A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Executive Director of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members, as well as the broader scientific community, about topics relating to antibody discovery and development. Our expert speaker, Elud Odo, will discuss accelerating antibody drug discovery through computational modeling. Please note, the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speaker.
Thank you very much, Jan, for the introduction, and also to the Antibody Society for hosting this webinar. I just want to start by saying that there really is no doubt in many people's minds around this webinar table that monoclonal antibodies are the most powerful tools, or one of the most powerful tools of modern medicine that's available for us, and has really radically changed the way we treat many diseases and even the way we prevent diseases. I was just looking yesterday at the statistics put together by the Antibody Society and saw that since 1985, there's more than 100 therapeutic monoclonal antibodies that have been approved, and about another 20 that are currently in review. And yet we all know that there's still yet many great medical needs that can be addressed by monoclonal antibody-based therapeutics. And besides that, there's also the issue of global access. Many people in the world still do not have access to these groundbreaking therapies. And so the question is, are there strategies that we can employ to try and accelerate the rate at which we are discovering and making available these great medications? And is there a role that computational modeling can play in aiding that process? And so that's what I'm going to talk about today in my presentation. And my goal really is to demonstrate that computational modeling continues to improve and get better and better at addressing problems, not only in small molecule drug discovery, but also in biologics. And I hope to show you that the right methods, if applied in the right context and used properly, can provide value. Value in terms of saving time, research effort, and also value in terms of saving money. And also, I'd like to assure you and hopefully get you more comfortable with the idea that you can, in fact, apply computational modeling. It is accessible. There's been lots of advances in the last few years to make these methods more generally available to practitioners in the field, people in the bench, for example. So I'll start by talking briefly about the role of computational modeling in antibody drug discovery in general, and then transition to talking about how do you get from sequence to the sequence to 3D structure. Once you have a structure, then the next thing you start thinking about is how to extract value from that structure, calculate properties from that structure. And ultimately, you get to the point where you want to consider optimization, engineering your antibody to make it better. And then I'll summarize just a forward-looking perspective on where computational modeling is headed, especially in relation to antibody drug discovery. So let's get started. So over the last few years, we've seen great advances in our ability to reliably model biological molecules. And this really began in the early 70s with quite considerable effort to better understand and parameterize molecular mechanics computationally. And that led to a lot of expectations, very high expectations. The idea then was that, oh yeah, computational modeling is going to really change in short order the way we discover drugs. And that turned out to be not the case earlier on. It was just a little premature for that. But we've gone through that season of disappointment, if you will. And since then, there's been quite steady progress in our understanding of molecular systems, in the way we can describe them computationally, and developing solutions that are much more robust than the ones we had in yesteryears, not only in terms of software, but also there's been a huge acceleration in the ability and capacity of modern hardware to handle some of these very complex systems, as you can see represented in this slide. And so what I would say is that although many difficult problems still exist, some of them out of the range of current molecular modeling capabilities, we are now at a point where we can confidently say that the technologies that have been developed over the years are beginning to make real-world impact on biologic drug discovery, and that includes antibody discovery. I think many of you will agree that antibody drug discovery is hard. You know, there's many steps involved in bringing antibody-based therapeutic from the bench side to the bedside. And along that pathway, there are considerable challenges to be overcome in order to achieve safety, efficacy, and as well as developability endpoints. And some of these challenges are unique to biologics and even unique to antibodies compared to small molecules, for instance. And I would say that not all of these stages are equally amenable to computational modeling. So the question becomes, where and when can computational modeling have the most impact? So if we look along this pipeline and going from left to right, we can see here that in the very early stages of target assessment, when you're just beginning your project, identifying what your target is, making sure that you have the right antigen and that it is suitable for generating your antibodies, what can you do with computational modeling? There's several things that you can approach this, the problem with. One is if you don't know the structure, there are very good tools and solutions out there for building structure from sequence, protein homology modeling, for instance. And if you even have a structure that's experimentally determined, a lot of the refinement of these structures depend on computational methods that have been developed over the years. Now, if you go a little bit further into hit generation and trying to identify your leads, then early developability assessment is very, very important. You don't want to carry a molecule far along the process, only to realize at some point that, oh no, you know, there's a problem here that could have been addressed earlier, or at least could have been detected and informed you to pick a different candidate, for instance. And so there are several tools that can address that, including early developability assessment. You could generate lots of descriptors from the properties you calculate on your structure and use those to make predictions as to what molecules have the best prospects going forward. And then ultimately you get to the point of having a lead that you want to optimize mutationally, mutational engineering, and that's an area that is very good and robust tools for doing that in the market today, and also in academia as well. So in summary, what are, if you were to look at any modern modeling software package, what did you expect to find? So you'd find a range of capabilities as listed here. You're going to find, hopefully, some solutions for structure prediction by homology. And once you predict the structure, to assess the quality of the structure and its reliability for downstream application. You also find solutions for structure refinement, improving the quality of the structure if there are certain issues that need to be addressed. In addition, there will likely be solutions for protein analysis. So these are the solutions that are present in the Schrödinger suite, but other software, comparable software, would have similar capabilities. And of course, we know that proteins and antibodies are not static; they are dynamic, and so being able to study the dynamic properties of your molecule is an important thing that you can do with software. Protein engineering, all set of solutions there, various levels of accuracy, and I'll talk about that later on. But specifically for antibody modeling, there are solutions that have been designed to address just that, antibody modeling. I'll talk about that as well. And of course, protein-protein interaction is a huge area of interest. Proteins do what they do by interacting with other proteins and other molecules in the cellular environment. So having all these capabilities is really nice, and the increasing availability is a very positive development in the field. But at the same time, it is important in using any solution to look at the scientific validation. Scientific validation is essential because it defines and informs the domain of applicability of each method. And so I would say that any measure, predictive method that you use in a piece of software warrants some kind of publication or paper, that is associated with the method and helps you to understand how the method was developed, how strong are the results in that paper, and importantly, what assumptions were made, what approximations were made in developing that method. The bottom line, though, is that each method has its strengths and weaknesses, and it's good to be aware of that and to try to identify those early on. Talking about the ability and accessibility of these computational methods, an important development in the field over the years has been professionally designed user interfaces that make it easy for someone who is not an expert in the field, someone who is a bench scientist who is curious about modeling and wants to see what value it can add to access these powerful methodologies in the back end. And you could have a method that performs very well, is very powerful, but if it's not accessible to people actually designing and developing antibodies, and it's difficult to run, then the impact of that great technology really is limited. And this is especially the case for biologics, where many companies do not have full-time trained modelers. In the small molecule field, that's more often the case. In biologics and antibody drug discovery, it's typically the crystallographer or the bench scientist, the molecular biologist who is interested in running some of these calculations. And so it's very important to have friendly user interfaces with reasonable default settings that make these solutions accessible. So transitioning to things from sequence to 3D structure, it is a fact that for most antibody programs or projects, the starting point for hypothesis generation and making predictions is usually primary sequence. And this could be a nucleic acid sequence or even an amino acid sequence. But as I mentioned before, antibodies are much more than two-dimensional linear entities. They have 3D structure, they are dynamic, they interact with the neighboring proteins and other components in their environment. And so it becomes very valuable to be able to have 3D structure information about the antibody that you're working on. And this could be in the form of a crystal structure, or a cryo-EM structure, an NMR structure. And there are many structures, 3D structures have been determined for proteins in general, and many continue to be solved. For antibodies in particular, there's approximately 7,000 antibody 3D structures in the PDB. This is cataloged well in the structural antibody database, SubDB, that's hosted by the Oxford Protein Informatics Group. So it is important to remember when you get a structure from the PDB or from whatever source that really these experimentally determined structures are in fact models. The models fit into experimental data, electron density, for instance, and it is important to treat them as such. And what do I mean by that? What I mean is that before you start using a structure, you need to ask some questions. You need to be very clear about the problem you're trying to solve. And for that problem, what level of accuracy or what level of detail is required in the structure to be able to address that problem? And what calculations are going to be running to tackle that problem? What are the required inputs for that calculation? Oftentimes, there's more than one PDB structure. If you're lucky, of your antibody of interest, for instance, you need to compare all of them and make sure that the one you pick as your starting point is the one that is the best quality, that will give you reliable results down the stream. This is important because if you start with a structure that is substandard or not the best that you could have started with, then that will be reflected in the kind of results that you get from your modeling. So to enable you to do that, there's several things you can look at in terms of structure quality, quality assessment. I'll not go through all the details, but generally a common thing that people look at is the resolution of the structure. And the lower, the better the resolution, the more detail you can see, as shown in this graphic, at different levels of resolution. And as the resolution gets worse, then even sidechain placement becomes uncertain. And so if you're going to do some calculation that requires that the sidechains are accurately placed, then you should be aware that the resolution is a factor to consider. But it's not really the only factor. There are many things. And these days, it's fairly straightforward with the help of computational software to be able to automate the analysis of the structure quality, assess the quality of your structure. And many of these solutions, this is just a screenshot from the Schrodinger implementation, gives you visual feedback that will tell you through these big red circles that this is a certain parameter that is off outside the range of expected. And so you should then take a closer look at that and see if it's an issue that can be addressed, or if you should go back and try to see if you can find a better structure. So it's fairly straightforward these days to look at these and be able to make the right decision. All right, so also about the case where you're lucky that you have a structure in the PDB, but more often than not, there's no experimentally determined 3D structure in any database. And so you are then in a situation where you need to predict the structure from sequence. And there's various ways for doing that. You can do that by homology modeling, or more recently, you can use machine learning, AlphaFold, for instance, to help you generate the structure of the protein that you're interested in. But the thing is that the technologies exist to help you build models. I would say, though, that, just like I said, for 3D structures determined experimentally, for homology models or even AlphaFold models as well, you really do need to do structure quality assessment to determine if that structure is good enough and the regions that you care about are resolved well enough for the kind of purpose that you want to use it for. All right, but talking specifically about antibody structure prediction, there are deliberately designed protocols for antibody 3D structure prediction based on homology. And from the data that I showed before, there's templates that exist, antibody templates for the framework and also for the loops. But there is one loop, the H3 loop, that requires special attention. And the reason is shown in the graphics that you see, the graphs that you see here, the plots that you see on the top right. So this really shows, if you look in the PDB and look at all the H1 loops and you align them, the H2s, the H3s, all the serial loops, and you try to see, how close are they to each other, what level of variability exists, structural variability, you can see that the H3 loop is very diverse structurally. And that makes it very much more difficult to predict the structure of computationally. Structure prediction by homology basically devolves into three key phases, I would say. The first is to model the framework regions. And once you model the framework regions, then the next step is to model the CDR loops. And the last step is to model the H3 loop more accurately, because that might require techniques and approaches that are different from the approaches that you use to model the other loops. So I would say that, H3 loop prediction, the accuracy of that really is what differentiates the different methods and the different software solutions out there. How well can they predict the H3 loop? The rest of the antibody, generally the performance, if you compare different prediction protocols, are quite comparable, quite similar. So let's look more closely at the H3 loop. And as I said, more robust techniques are required to model the H3 loop. And the reason is that there's no simple rules that apply to the H3 loop. The other loops have canonical conformations that can be classified. And so if your particular sequence falls in a certain canonical conformation, then the prediction becomes easier. But that's not the case with the H3 loop. So some of the methods in the Schrödinger suite, for instance, we use an approach called prime, which is really de novo loop prediction based really on physics, just looking at the physical interactions and taking care of all the details of the molecular mechanics to predict the CDR H3 loop. And this is validated in a paper that I show here on the right-hand side, where we looked at about 3 structures in a benchmark data set. And that data set consisted of some short loops, some really long loops. And the prediction had 2 different pathways. One was if you're given the structure of an antibody, the crystal structure, and they just ask to predict the CDRH3 loop, how well can you do that? But what if you have a homology model and then you want to predict the CDRH3 loop based onto that homology model? How well can you do that, and what's the difference? And the results are shown in the next table here. And what you can see here is on the left is the various different kinds of loops, different entries in this data set, the loop lengths, the variability in loop lengths, you can see there. And then if you just look at the database in the PDB and see, which is the best loop, if you just to pick, not based on sequence, but just the loop that closest to the true crystal structure that's known, how close is it? So the numbers here you see best in database is the RMSD, root mean square deviation. How much does it deviate from the true conformation? And so the lower the RMSD means the closer it is to the known structure. And so in the next 2 main panels here, you can see using crystal structure. That's what I mentioned before. If you're trying to predict the loop in the context of a crystal structure, just the H3 loop, the rest of it. And then on the right 2 columns, if you have a homology model. I think the main message here is that if you just predict based on homology modeling, you can see that those predictions generally worse than the prediction that you get using the de novo loop prediction approach, which is highlighted here in a yellow box. You can see that the RMSD is generally lower, whether you're using prediction of H3 from the crystal structure or you're predicting it in the context of a homology model. And so it really emphasizes the point that, typical homology modeling approaches, the standard doesn't work. You need to do something special to get a good prediction of the H3 loop. All right, so having gone through all that process and you have a 3D structure, then now what can you do with it? And this is really where the fun begins, because now that opens a whole world of possibilities in the world of modeling, in the sense that you can now start querying things like, all right, so I have the 3D structure. What are the dynamic properties of it? In that case, you might want to use molecular dynamic simulation, for instance, to see how the conformation evolves over time. You could also calculate protein properties based on the structure, or look at protein-protein interactions, or use that as the basis for making mutations during protein engineering to optimize a particular property, or in case, for instance, of humanization of an antibody, do your CDR grafting based on the structure. And that's the reason I was emphasizing before that, you have to make sure that you get the best possible structure you can get, whether it's a homology model or crystal structure, in order to be successful in doing any of these protocols that are listed here. Now let's start with protein property analysis. What kind of properties can you really analyze? And there are many of them. We can't cover all of them, but I just wanted to list some of them that are available here. There's various kinds of descriptors that you can calculate given a structure. Some of them actually don't really require structure, they're purely sequence-based. So there are what we call scalar descriptors, things like pi, net charge, electrophoretic mobility, radius of gyration, and then there's purely sequence-based descriptors that you can calculate, as well as, and importantly for antibodies, region-specific descriptors. In other words, descriptors that are not just for the whole antibody, but more granularized in the sense that you can calculate properties for a particular, framework region, a particular CDR loop, H3, H1, H2, and be able to really zero in if you're observing a certain characteristic, being able to zero in on a particular part of the antibody that's most responsible for what you're observing. One of the properties that you can calculate that is not included here, that is included here is aggregation scoring. Now, this is one of the methods that is implemented in the Schrödinger suite, whereby given a particular protein, you can calculate its propensity to aggregate. And that you can do by generating a surface profile like the ones shown in this figure here. And the unique thing about this is that I think most aggregation prediction methods look primarily at hydrophobicity, but these particular methods looks not only at hydrophobicity, but charge as well. The premise being that if you have a hydrophobic patch that is surrounded by polar or charged residues, then that really does impact the contribution of that particular region to aggregation. But beyond looking at surfaces, you could also calculate a numerical score called AG score, and that is useful for ranking. For instance, in this blinded study of 5 FVs, calculating AG score and then sorting them by AG score turns out to perfectly rank them in order of aggregation risk, with the highest scoring one being the one that has highest risk of aggregation. So this method actually has been validated in a number of studies, and there's publications out there, not only of the development of the method, but also the application of the method. As I showed before, there's many descriptors. This is just one of them, but something else that you can do with the descriptors is to generate QSUM models. So if you generate QSUM models and look at the regression plots for some of these, and in this case, as an example, looking specifically at about 97 integrin binding monoclonal antibodies and seeing if we can predict HIC data, HIC rotation times here. And not only can you generate these QSUM models, but I would say most importantly, they can help you to identify descriptor importance. In other words, the descriptors that are the key drivers for this prediction, the key influences of the prediction of retention times here. And what you can see here, it really does make sense. AG score is one of them, but you can also see, for instance, the H3 hydrophobic patch energy, for instance. So this is all available. I think a lot of people are interested now in machine learning and using the descriptors. The only question I'll provide is that quite often people don't have enough data to run some of these models reliably. And in those cases, I think it's good just to do a QSUR analysis and try to identify descriptors that drive the property of interest, and then use that as a launching step for hypothesis generation in terms of what can you do about addressing that problem, whether you need to engineer or to do something else. With the structure 2, opens the world of antibody-antigen docking, or any other kind of protein-protein docking. And for those who are not familiar with protein-protein docking, really it's a search and score protocol. Basically, you're starting with 2 potential interacting proteins and reorienting them in many different ways, and each time testing contacts and scoring them based on energy. The idea is that you're exploring this energy landscape, trying to find the interactions or the poses, as they are called, that are low energy. And so the low energy poses are presumed to be the ones that are most likely to exist. Now, to really do this effectively, there's a few things you need to know. First, you have to have a good structure. And then you need really to have, if you have some experimental data that gives you some idea of where the interactions might take place, whether it's mutational data or anything like that, it is really, really crucial to incorporate that into your docking calculation. Otherwise, things can become very, very difficult, especially if you're doing totally blind docking where you don't know which part at all of one protein interacts with which part of the other protein. But the scoring is all based on an energy function of the type shown here. I'm not going to go into the details. I just want to walk you through the steps of a docking exercise. So most docking, not all of them, but most docking protocols in the market today are rigid body search. In other words, conformational flexibility is either not accounted for or accounted for minimally. And that's really comes down to just the complexity of accounting for all possible conformational changes and also for the docking as well. But there are, flexible docking methodologies out there as well. And so basically, you do the rigid body search, searching for the poses. And then once you do that, then you end up really with a large number of potential interaction orientations. In this particular case, using Piper, 70,000 poses that you end up with. And then now you have to filter through these and select which ones make the most sense based on energy scoring. And once you do that, at the end of it, you end up with just 1,000 low energy poses. And this has been found to work pretty well in most cases. And this has been shown in Capri competitions. So this approach works well with Piper. But then once you have those 1,000 poses, still you have the task of, which is the one that really represents the truth. And that's a really difficult determination to make. And one way to do that is you take the 1,000 and cluster those conformations. And once you do the clustering, then you end up with a few clusters and then look at the centroids of each cluster as representative of that cluster. And you end up with about 10 to 30 centroids. Now then, by ranking of these, the most difficult problem really here is you can narrow down to top 10. Usually the top 10 contains oftentimes the true, experimentally validated interaction interface or pose. But then picking that one from the top 10 is still a fairly difficult problem. And that's what I was alluding to when I said if you have any orthogonal data, experimental data, or some information that helps you to figure out which of those is most likely the correct one, then that really increases your chances of success. All right, so I'll just give an example of a case study. So this is case study of PD-1 binding to this antibody. Two antibodies, in fact, nivolumab and pembrolizumab. Those two, there are crystal structures of these in complex, these antibody antigen complexes. And it is known that nivolumab recognizes the FG loop of PD-1, and similarly, pembrolizumab recognizes the CD loop it dominates the interaction between those two. So then what we wanted to do in this exercise was really to ask the question of, can we, if we only had the sequence of pembrolizumab, for instance, and we wanted to predict the interaction interface with PD-1, how well can we do that computationally? So the first step, of course, is to build homology models from the sequence of the antibody, predict the structure using homology, using all the protocols that I showed you before for predicting the framework region, and then the loops, and then comparing that, first of all, to the non-crystal structure. And what you can see here is that the predictions are pretty good. There's a little bit of a misprediction in the H1 loop here, but otherwise, for both antibodies, the predictions are quite close to experiment. Now, the one little detail that I didn't mention that kind of makes things complicated is the fact that it is known that the city loop of PD-1 is disordered. In other words, the crystal structure will not resolve it and show you how that loop looks. It is stabilized, however, when it binds to one of these antibodies, pembrolizumab here on the right. But if you're going to do a docking exercise, you're most likely starting with PD-1 without that loop. And so we also know that, the conformation of that loop is hard to determine, it's kind of uncertain, and it really complicates things. So the question is, can we actually be able to make that prediction and come up with a reliable prediction of what the complex would look like? And so that's what we went about doing here. The first thing you have to do, of course, is to predict the conformation of that disordered loop. And this you can do computationally through loop sampling. And basically what you end up with is many possible conformations of that loop, but you don't know which one, in fact, is the one that binds to your antibody. And that's, of course, the problem we're trying to solve. And then once you do conformational sampling of that CD loop, and then you use your predictor structures of the antibody itself, and then you do docking to all these possible conformations of the CD loop, and then you're left with the task of figuring out which of those predictions is the best. And that's where your orthogonal data comes in, as I said before. And what could that be, for instance? For instance, we know in this case that there is a mutation that abolishes binding to human PD-1. And similarly, by comparing species, we know that this mAb displays low or worse binding towards mouse than to human. And so the question then is, what's the differences between mouse and human, and what does that tell us about where the binding might take place? So that's precisely what was done in this study. As I mentioned before, loop modeling, especially some of these really difficult loops, require a little bit of a more robust computational approach. There's quite a bit of heavy lifting involved, and so you got to pull all the stops and maybe use really powerful techniques to do this. And one of these is called REST sampling, REST replica exchange molecular dynamics. Without going to the details, I could really simply describe it as what we're doing here is essentially enhancing sampling of the loop by increasing the effective temperature around the region of that loop, heating the loop, if you will. And then once you do that, we end up with possible conformations. Again, we cluster those loop conformations and take representatives from each cluster. And so what we do then is the homology model that we built of the antibody. We take that and then we dock it to the top representatives of the PD-1 loop predictions that I described before. And then once you do that docking, then we end up with many poses. And then now the orthogonal data comes in the sense that if we compare the sequences of human and mouse PD-1 here, we can see that there are about 12 positions, and we can map those onto the structure. And then we can now filter the poses that were generated computationally and see which ones really meet this criteria where these regions are most likely involved in the interface. And if we do that kind of filtering really from the many thousands of docked conformations that we got, we end up with just really 2 that fit that criteria. And if you look at the statistics for those 2 predicted docked poses, you'll see that one of them, the top one here, number dock P29 here, has much better parameters here in terms of, if you compare it to the experiment, you see it has quite similar to the known structure of that complex. And besides that, if you even didn't know the structure of the complex, you can see that other parameters like the number of hydrogen bonds that it forms at the interface, a pretty good number, more than the other choice you have of these 2, and it makes a very good surface contact area. And so that seems to be one that makes the most sense to pick as the final prediction. And if you do that, and then now you compare your final prediction chosen that way with experiment, because as I mentioned before, this was a retrospective validation exercise where we had the experimental complex. We wanted to see how well we could predict it in spite of the challenges with the CD loop. And you can see here that the prediction here is pretty very good agreement with the experiment. The interaction with the interface are very well reproduced. And so it was quite interesting because, as I said, docking is really challenging. And if you have uncertainties like where certain loops exist, an H3 loop or even CD loop, then that makes things a little bit harder. But what you can see here is that if you use advanced computational approaches and you marry that with orthogonal data, then your chances of success are quite good, reasonable. And so if you do this kind of exercise on a project, you of course then want to say, okay, this is my predicted pose, feeling very confident with it. Maybe you can make a few more mutations that are predicted to be the interface here and see if that has an impact on binding, just to validate. And once you have that kind of validation, then now the possibility of doing engineering, mutational engineering for your project, for optimization and other things is opened up to you that way. All right, so we've gone through predicting interfaces. I wanted to talk about protein engineering in terms of engineering antibodies for stability or affinity, which is a very common need. I mean, quite often, for instance, for stability, even if your goal really is not to enhance stability in terms of melting temperature, but maybe you're making a mutation to address aggregation, you want to make sure that that mutation doesn't compromise other properties like affinity and stability. So there's lots of multi-parameter optimization that is involved in antibody discovery, as you all know. So the question then is, what is the standard approach right now? I think quite often now, experimentally, if you want to do candidate optimization, you're looking at either directed evolution using phage display or yeast display or some other technique. Or you could just do site-directed mutagenesis if you have some idea of where you want to target your mutations and explore the impact of those on the property of interest, whether it's affinity, stability, or something else. If you do that, that works, but it also takes a lot of time and it can be quite expensive. And so the question is there anything that computational approaches can offer, and how reliable are they for this kind of application? And so what I'm showing here is computationally driven workflow that we have, whereby if you want to optimize certain positions, you identify positions to enumerate. Either you can identify those positions computationally. If you don't know where they are, there's several modeling approaches that can help you on where to direct your focus. Or if you have some knowledge from your work with that particular antibody, then you might have some idea where you want to focus on. But anyways, the basis of this really is that the first step is really a very rapid approach for essentially classification using this approach that I call MMGBSA. And basically here helps you to throw away the candidates that are not worth expanding experimental time and effort on, and then have just the top set. So the good and the bad, essentially, that's the level of resolution you get with this approach. And then once you make that cut, then now you can apply more rigorous approaches like free energy perturbation, which is a molecular dynamics-based approach using very accurate representation. Well, not very accurate, but really detailed accounting of the atomic detail of your system, and especially water, compared, for instance, to MMGBSA, where water is modeled implicitly. So in other words, a rough cut at the beginning, and then a more accurate cut, and then you end up with a small set of mutations that you can then test or validate experimentally. So in other words, computation here is helping you to really narrow down your focus to where you have the biggest chances of success in terms of what mutations to make. Now, so how does that compare? So in one particular project where we did this, we're just looking to see, what kind of success rate can you expect? So in this case, it was actually antibody-antigen complex. And as you go, the first approach on the left here is a computationally driven workflow that I just showed you. And you can see that what we were able to do here was to identify the 10 top ranked positives in about 3 weeks. So in this exercise, there was parallel experimentation that went into making mutations and testing, for this whole system. About 930 different mutations were tested experimentally. And we're just trying to see if we had used a computational approach, what would that mean? So 10 positives in about 3 weeks. If you had to maybe go to a CRO and help them, ask them to do the same level of scanning for all those positions, it would take maybe 6 months roughly. And if you do the direct evolution approach, you're looking at about the same amount of time and maybe more, depending on how many cycles you have to go through. So the idea here is that, you could save quite a bit of time and get results faster and move your project faster by adopting computational approach. So I talked about two approaches, the MMG-BAC approach. I'm not going to go into great detail, just to say that, it is fast, and oftentimes it's a trade-off, speed and accuracy. Oftentimes, when you gain speed, you might have to make some assumptions or approximations that compromise accuracy, and also the reverse is true. And so, but there are times when you need speed and times when you really accuracy is the most important thing as you get closer to narrowing down to the candidate of choice. And then I talked about protein FEP. This is the molecular dynamics-based approach, which is more robust, very accurate in a number of systems that we've looked at, but takes a little bit more effort in terms of computational time. And here are some of the initial studies that we did using FEP, just looking at GP120 and looking at binding affinity change upon making a mutation. These are really alanine mutations. And as you can see here, the root mean square error tells you how close you are to the actual experimentally measured change in binding affinity. And so the lower the error, the better the prediction. And you can see that the R squared on the left, the regression here was pretty good, about 0.62 for FEP. In this case, we divided the data set into two. There were some mutations that were close to glycan. And this just shows some of the challenges that still exist in modeling. Modeling these sugars can be pretty complicated. And so the accuracy takes a little bit of a hit if you are considering the cases where the mutation was made close to a glycan. Similarly, using the same FEP approach, this is a study that was done by a group at Merck, and they were trying to see this for thermal stability, where they had melting temperature data on a set of proteins from the Protham database. As you can see here, that again, if you compare FEP approach, you get a better prediction accuracy compared to some of the most standard approaches. And on a larger scale, a more recent study that we did was where we took a data set, a well-curated data set curated by this Pushy and company in a study. And this was about 328 single-point mutations. And we looked at these and tried to see how well does FEP predict the delta Gs. And on the left is FEP prediction. On the right is using just the standard approach, the MMG-BAC approach alone. And you can see that for the extra detail that FEP incorporates, you get better predictions. Now, the question that comes up, of course, then is, what if these were based on crystal structures? What if you just have homology models? And there's a recent study. This is still in bioRxiv. It's a preprint. And we compared crystal structures versus homology models. And the summary here is that you can see that, if you use homology models instead of crystal structures for the same system, and you build your homology models well, then the hit inaccuracy is not as great as you might have feared. You can still get reliable results using homology models. So in summary, I just wanted to say that computationally driven approaches are offering new and quite powerful and targeted ways of helping you engineer antibodies, optimize your antibodies, and focus your effort where it's most likely to pay off. At the same time, it's very important to make sure that you're using the appropriate technology for the kind of problem you're trying to solve, and not just using solutions without insight into how they've been developed and the assumptions that have been made. So from the results I've shown you, I hope you can see that current results are quite encouraging for prospective applications for things like stability and affinity and selectivity and many more things to come. And finally, as I said, I think these methods are accessible, and I think a lot of people in the industry now are getting to use computational modeling. But if you're wondering how to get started, maybe you're a bench scientist, you want to learn more, we have this course called Introduction to Computational Antibody Engineering. And this course, there's an upcoming session on November 7th, which you can enroll. And there you learn a lot of things about how to get started in antibody engineering and practical steps related to some of the things that I've discussed in this presentation. And with that, I just want to end by saying thank you very much. And I'm happy to take any questions.
Excellent. Thank you very much. And yes, indeed, you do have a few questions. For our first question, they're coming in so quickly, I have to shift my screen around. First question, what is the source of the benchmarking data set of 53H3 loops? Is the reference included?
Yeah, the reference is, I can provide that. I think I showed the paper that describes the study for benchmarking those loops. And within that paper, there's a reference to the source of the data. So I can provide that later on, hopefully along with some of the slides, although you'll have to confirm that, yeah.
Okay, that's fine. I did answer a question related to that, but I'll also verbally say that everyone who registered will get a link to the on-demand version. And the PDF, the slides that he's talking about, I'll turn into a PDF, and it will be attached to the on-demand version. So you will have access to that, including the reference he's talking about. Next question. How different is protein structure analysis from protein reliability analysis?
Yeah, I think those are related. I think you analyze the structure in order to determine how reliable it is. And so for protein reliability analysis that I showed here, really it's just a visual representation of the outcome of protein structure analysis. You're looking at things that might be off. By off, I mean things that are outside of the standard parameters statistically analyzed from the PDB database, for instance. And so the report just tells you, yeah, this is where to look. But essentially what you're doing is protein structure analysis in order to determine the reliability. So they're very closely related.
Next question, how big is the human sequence database for antibodies? Is a secondary question to that. Is there a mouse sequence database if I want to do murrinization instead of humanization?
Yes, I believe those databases exist. I don't know how big they are. I would imagine very big. I know there is the antibody repertoire database, I believe, that's hosted by the Oxford Protein Informatics Group, I think. Yeah, but I don't know how big those databases are. But I think you can find them if you look them up. But I would imagine that the database of antibody sequences is pretty huge, would be my expectation.
And for those who are interested in resources like that and links to them, I'll just take a moment and direct you to the Antibody Society's website, because we have a whole page dedicated to links for such resources.
Awesome.
Nice work slipping that one in there, Eliud. How can we take into account CDR loops flexibility during protein-protein docking? Secondary question. Or do we need to know beforehand how the loops are placed in the bound conformation to get good results?
Yeah, I think, if you know how the loop is placed in the bound conformation, then, that's an advantage that oftentimes you don't have. So, in this case, what would be best would be to sample the possible conformations of the loop, as I showed. The example I showed was where we are doing thorough loop conformational sampling on PD1, which is the antigen. But you could do the same thing for the H3 loop, for instance. And then what you end up with is an ensemble of predicted H3 loop conformations for your antibody. And you could then cluster. I mean, there'll be very many in that ensemble. So you can narrow that down by clustering and just taking the centroids of each of those clusters and then using those for docking to your antigen. And that's how you'd handle that. But again, as I said, it would be good if you have some information that will help you to fish out the right dock, or the most probably right dock conformation from that ensemble. Because you can imagine, even if you're just starting with a specific, fixed loop conformation and you're doing docking, that docking exercise itself, as I showed, you end up with 70,000 different dock poses, which you can filter down to 1,000, and which you can then further filter down to 30. And that's just for one specific loop conformation. If you have an ensemble of CDRA3 loop, then you multiply that by whatever the number of predicted possible conformations of your CDRH3 loop. But essentially, it's ensemble docking. And then you have to have a good filtering criteria for determining which is the most likely result. It is computationally expensive, I have to say. But I think with increase in computational power, and you have the cloud, and you can really parallelize these calculations and just run them all.
Next question, why do we exclude cysteines?
I'm not sure what the context of that is.
Well, it came in at 11 o'clock and 47 minutes into your presentation. So I don't know if that gives you a clue. It was fairly late in the game.
All right. Yeah, I suspect there might be. Okay. Yeah, I'd have to guess where that might be. But I'd say, cysteines. Oh, yes. Okay. Now I get it. Yeah. So this is where I was talking about looking at protein stability and affinity based on some data set. And I was showing the timelines, I believe, and saying that we excluded cysteines. Yeah. So, I mean, the idea is that, cysteines typically are involved in some kind of disulfide bridge. Oftentimes, you might have some free ones, but we just didn't want to mess with that, So in terms of identifying positions to mutate, we try to leave cysteines alone because they're there for a reason, unless our goal really is to design the sulfide bridges or to free them up for whatever reason. So that's really was just a choice in terms of where do we want to make the mutations. There wasn't any other reason. Yeah.
Okay, well, we have one final question that I could actually make a few comments on myself, because it's how much does the course cost? It's actually quite affordable. I don't remember the exact number, and Ellie, maybe you can jump in with it, but as I recall, it's cheaper for students and a little bit more expensive for everybody else, but on the order of $300 or $400. But I can also take this opportunity to remark that if you're an Antibody Society member, you get a 10% discount. And if you want some information about how to access that discount, then go to the members only section of the website, where you will find a whole page dedicated to discount codes. Elu, do you want to add any more to that?
Not much more, but I just say that for more information about the cost and the sessions and even the content of the course, I would direct you to the website that I'm showing on the screen here, schrodinger.com, and then Introduction Computational Antibody Engineering, and there you'll find all the information. Or you could just do a Google search, and that will direct you to the Schrodinger page that describes the course very well.
Well, excellent. Thank you so much. I think we can wrap it up at this point. So in concluding, I'd like to thank our speaker for providing insights on how to accelerate antibody drug discovery through computational modeling. And I thank you very much for joining us today. An on-demand version will be available in a few days. I will send that link to everybody who registered. Please do feel free to watch this or any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.
Thank you.